Welcome back. In this lecture, I'm going to finish up on the carbohydrates, and we have one group left. This is oftentimes what we call the complex carbohydrates. These are the polysaccharides. Poly, as you know, means many. Saccharides means sugars. So these are generally ones that have more than two monomers. We will see that there is a smaller group that have 3 and 4. These are called oligosaccharides, but I'm going to show you a couple of those at the end of the lecture. But generally, when we think about polysaccharides, anything that's more than two monomers or 2 monosaccharides in the polymer. The polysaccharides we're going to talk about, several that come from plants, one that's found in us and animals, and then one that is found in arthropods and also in fungi. So let's begin. The first one I want to talk about is a common form of plant starch. As you know, we get most of our starches from seeds. Seeds, as you know, are the lunch that the mama plant packed for the baby embryo. Every seed has a little embryo in it. It's got some proteins, it's got some oils, and it has a lot of starch. And there's 2 major types of starches that are found in plants. The simplest type is this one right here called amylose. Amylose is very, very simple in that it has just one type of monosaccharide. In fact, this is just nothing but alpha glucose. You'll notice that it's just like maltose, except that it's longer. So the bonds that connect one alpha glucose to another is an alpha because it's alpha glucose, 1 to 4 because it bonds between carbon number 1 and carbon number 4. And since it's a covalent bond joining 2 monosaccharides together, we call it a glycosidic linkage. So, same type that we found in maltose. Now, if you're wondering about the name amylose, right, amylo comes from, can be translated as starch. So this is literally the starch carbohydrate. That's where it gets its name from. You might know that in our saliva, we have an enzyme that breaks this down. A salivary enzyme that's called amylase. Remember that ASC often tells us it's an enzyme, and enzymes are often named for what they work on. And this one uses some hydrolysis to break down amylose into smaller bits. Now, in a typical plant starch, we're going to find that about 20% of the starch that's stored in plants is in this form, in the amylose form. So, amylose, just alpha glucose, all right, it's a polysaccharide, and the chemical bond that joins them together is an alpha 1 to 4 glycosidic linkage. Now, turns out that amylose, because of a lot of its hydroxyl groups, is very attractive to water, as a lot of carbohydrates are. They're very soluble in water. Amylose does a very strange thing when you place it in water. And let me show you this little graphic here. It turns into a helical structure. You can see it here. So it makes this little spiral. And as you can see, we have, a front end and a hind end, so very similar to what we find. So that is amylose. It's of our starches, it's the minor one. We're going to now look at the starch that is the most common and the most abundant. It's very similar to amylose, but we'll find out that it is actually a lot more abundant because it has branches on it. So let's begin and take a look at our next one, which is called amylopectin. So here's amylopectin. It's still built from the exact same monomer. This is all alpha glucose. So you can see it's just a long chain of alpha glucose. The interesting thing about amylopectin is that it has not only the alpha 1 to 4 glycosidic linkages, but it also has a new linkage, and that is between carbon number 1 and carbon number 6. Right? This one actually goes and forms the little side branch. Now, how often does this branching occur? It turns out on an average about every 25 monomers. So about every 25 monomers, there'll be a branch. Right? Now, the cool thing about amylopectin is the branches will also have branches. So this isn't just a single branch that comes off. The branches will also have branches. Now, there's a benefit for a plant storing starch in this way. By branching it, it can actually increase the number of alpha glucose molecules per unit area. You get a much more dense molecule. And this is perfect for storage. So if we think about where we find starch in plants, we're going to find it primarily in some place for storage. And that's a great one they're looking at seeds. So 2 bonds for it, alpha 1 to 4 glycosidic linkage, and a branching bond, alpha 1 to 6 glycosidic linkage. And again, the branching occurs about 1 out of every 25. Now I love this little picture here of amylopectin, and you can see that the branches have the branches have the branches. This also makes it very easy to add new alpha glucose molecules to the chain. Instead of just having 2 ends, a front end and a hind end, now you have multiple ends that you can add alpha glucose to. In the same way, if you think about adding, you can also go and remove alpha glucoses very quickly. Instead of just plucking one off one end and one off the other, and that's your only choices, an organism can remove quite a few of them at the same time using several copies of the same enzyme. Now the name amylopectin is an interesting one. Amylo, as you know, means starch, and pectin comes from a word that means congealed. So the congealed starch. Right. Now, our next type of starch is very similar to amylopectin, but in this case, it is different in that it is found in animals. Right. So the first 2 we've talked about are called plant starches. This last one is what we call an animal starch. And this one we've seen before. This is, oh, here's a nice little picture. I'm sorry, I jumped ahead of myself. This is what a potato looks like underneath a microscope. And you can see in green the cell wall. So you see the little outline of the cell walls. Those are the potato cells. And inside you see these oval-shaped bodies. All those oval-shaped bodies are starch granules. So you can actually go and see them. Now, they show up really well when you add a little bit of stain to it. We add a little iodine solution that turns the starch a blue-black color. And as you can see, some of the starch bodies there, these little granules, are a little bit blue, a little bit purple. And we do that so they actually go and show up. Iodine is a great indicator for the presence of starch, particularly what we call potassium iodine. And it's a great indicator for something has starch in it. If we compared, for example, a potato with lots of starch to maybe an onion, we would find that the potato has a lot more starch than the onions. The carbohydrates in an onion are mostly going to be stored as sugars. Alrighty. Now, as I mentioned before, we have an animal starch, and this is called glycogen. All right. Glycogen has a great translation. Anytime you see gen, it has several different ones. Think about the word genesis, which means an origin or a beginning. All right. We can also, I had a medical terminology book for years that used to describe it as agent that initiates. Right? So glyco means sugar or sweet. So the agent that initiates the sweet or the sugar. Or you can think of it as the origin of blood sugar. Right? Now, just like in our potato, where we're actually seeing the little starch granules, you can see little starch granules in a liver cell. That picture that you see up above, that big circular structure, is a nucleus. And to the right, you actually see lots of little black dots. Those black dots are granules filled with animal starch. They're made of glycogen. Glycogen, in a very similar way, looks a lot like amylopectin. It has, for example, these branches. So you have two types of bonds here. Everything that's in glycogen is just one type of monosaccharide. It's alpha glucose, just nothing but alpha glucose. Alpha glucose is bonded together by an alpha 1 to 4 glycosidic linkage. And occasionally you'll have a side branch. And you'll notice our little side branch again is going to be a bond between carbon number 1 and carbon number 6. So this is described as an alpha 1 to 6 glycosidic linkage. Now the big difference between this and amylopectin is that in amylopectin we had a branching of every 25th alpha glucose. We had a branch. Turns out it's much more compact in glycogen. Instead of 1 out of every 25th, it's every 10th one. So every 10th monosaccharide, we have a branch that goes off. And what it produces is a much more compact molecule where you have a much higher density. And, like we mentioned before, because you have so many branches, it's very easy to go and add new alpha glucose or remove alpha glucose molecules individually when needed. So let's take a look at sort of a macro picture of what glycogen looks like. And as you can see, it is incredibly branched. It has a lot of end. It has a lot of density. And this makes sense, especially for animals that have to sort of move around. You know, amylose and amylopectin work well for organisms that don't move much, but glycogen works very well and much more compact and more concentrated for for critters that move like animals. Now, turns out glycogen, as we mentioned before, we have about a 24-hour supply that is stored in our liver. And we have an interesting little dynamic relationship between blood sugar or glucose in the bloodstream and the amount of glycogen that's in the liver and the skeletal muscles. So I want to show you this little feedback mechanism that's regulated by two hormones. You've heard of one. This hormone is called insulin, but you probably have never heard the other one, which is another hormone called glucagon. When we eat a meal, right, and we start absorbing the nutrients, particularly fats and proteins and particular carbohydrates, as we digest these materials, our blood sugar actually goes and rises. Right? So we get a little peak in blood sugar when we go and eat. Now, blood sugar tries to be maintained within a fairly narrow range. As you can see, on average, we have a blood sugar pretty much at any given time around 90. That's 90 milligrams of glucose for every 100 milliliters of blood. Now, when we eat, our blood sugar rises. And so we have a hormone that reduces blood sugar. And it is secreted by a gland called the pancreas. The pancreas is right near your stomach, just behind your stomach, just a little bit lower, near the first part of your small intestine. The pancreas has these special cells, and they're called beta cells. And these beta cells actually go and detect when blood sugar gets high. When blood sugar gets high, then these cells secrete insulin. Right? Now, you might be wondering about the word insulin. Right? Insulin comes from a word that means island. It's the island protein. Anytime you see that IN at the end of the word, it usually refers to a protein. Right? So this means island protein. You might be thinking, island protein, what does that mean? Turns out that the cells that make insulin are found in a little island that is surrounded by other cells that make digestive enzymes and release bicarbonate ions. So the parts of the pancreas that actually go and make insulin and glucagon are found in these tiny little islets. So literally this is the protein, well, which is a hormone, that comes from the islets. Right? In the old days, we used to call them the islets of Langerhans. Now we try to get away from naming things after people. We call that an eponym. So we've been trying to get away from eponyms. Now they're called pancreatic islets. So that's where the word insulin comes from. Now, as you know, in order for sugar to get into your cells, right, it needs a little key. And that key is insulin. Insulin unlocks the cell so that blood sugar can enter into the cell. So what insulin does is by allowing the passage of insulin into the cell, it reduces blood sugar. So that's what insulin does. It reduces blood sugar. Right? Now, let's say that you skip lunch today. Right? So you skip lunch today, and it becomes about 3 or 4 o'clock, and you're feeling a little peckish. Your blood sugar starts to dip a little bit. Then what happens is another type of cell within the pancreatic islets, what's called an alpha cell, detects that drop in blood sugar. And it secretes a hormone, another protein that's called glucagon. Right? Glucagon. All right. Now, in glucagon, in this case, agon means against. All right. So this is against sugar. And what this means is this glucagon goes to your liver and stimulates the liver to start breaking off glycogen. All right. Now, I forgot to mention what happens to the excess amount of glucose after you eat. So your cells take up the glucose and the blood sugar, but let's say we still have excess blood sugar. That excess blood sugar then gets picked up by the liver and the skeletal muscles and converted into glycogen. Right? So that excess blood sugar after we eat goes and gets stored as glycogen in our liver and our muscles. Right? Now, when we don't eat, let's say we skip a meal, right, and our blood sugar starts to drop, that signals the the alpha cells to go and start secreting glucagon. Glucagon will go to the liver and stimulate the breakdown of glycogen. It'll start breaking off those alpha glucose molecules and releasing to the blood. So, what glucagon does is actually raise blood sugar. Now, as I mentioned before, we have about a 24-hour supply. So if you fast for 24 hours, your glycogen stores are declined or decimated, right? No more left. So where do they come from after that? Well, you have to go and start burning fat and proteins. All right? So we have a 24-hour supply of glycogen. Now, so far we've talked about some types of glucose that are digestible in living organisms, and that's going to be alpha glucose. Right? So we've seen it everywhere. Right? It's a component of all our disaccharides. It's the component in the plant starches and animal starch. Now I'm going to go and show you two polysaccharides that instead of using the digestible alpha glucose, uses the indigestible beta glucose. And if you recall, the big difference there is at carbon number 1. In alpha glucose, you have a hydrogen on top and a hydroxyl group below. In beta, it's flipped. We have a hydroxyl group above and a hydrogen below. That makes all the difference. So we're going to talk about two polysaccharides, one found in plants and one found in fungi and arthropods, that are indigestible to most things. There are some organisms that can digest it. They usually have a group of special enzymes that allow them to do it, but for most critters, it's going to be indigestible. So the first one is one that makes up the cell wall of plant cells, the cell walls of algae. And this is a type of polysaccharide that is built from nothing but beta glucose. It's just one long strand of beta glucose. And this is cellulose. Right? Again, the name comes from the cell wall of plants and the cell wall of algae. All right? So this is the sugar of the cell wall, the sugar of the cell. Now, you'll notice something a little bit odd about it. Right? It is unbranched. Right? There's no branches in cellulose. In fact, I love the graphic below because it kind of shows you an increase in scale. So you can see the individual glucose fibers. So here we start off with plant cells. If you look at a cell wall, you'll notice that it looks a lot like a textile. You've got the warp and the weave. You've got the up and down. If you go and you look at those individual little fibrils, you'll notice that they're made from tiny fibrils, and those tiny microfibrils are built from just long, long strands of cellulose. Cellulose is just this long chain of beta-glucose. Now, in this case, this is indigestible, but it's very useful for us. We call this dietary fiber. When we eat plants, right, particular ones that have a lot of cellulose in them, right, we do get a benefit, but it's not a benefit in energy. It's a benefit in a bowel movement. Right? Cellulose, which we call dietary fiber, all right, when it gets, it is indigestible, so it'll pass through your small intestine. It'll get to your colon, your large intestine, and it does a very important thing there. Right? It increases the bulk and the size of the feces. So as the feces are moving through, it's nice and big. The other important thing is because they're nice and big, they're easier to move. The other thing, too, is it makes the feces softer. And also goes and helps hold a little bit of water. You can see some hydroxyl groups there. Right? So you get these nice, big, firm, but soft feces, which actually makes it easier for the muscles to go and move the feces through the large intestine and then out to defecation. It makes defecation much, much easier. People who have diets that are very low in dietary fiber often suffer from constipation. Right? Constipation, usually when you have a low amount of fiber in your diet, then your feces are going to be smaller and more compact. All right? They're going to be harder, which makes it more difficult for the muscles to push against them. And because they're small and hard, they can often go and cause an outpocketing to form inside the colon. We call these diverticuli or diverticula. An individual one is called a diverticulum. They can retain feces, compact feces, and they become swollen and inflamed and particularly life-threatening. They can actually go and break through, and then we get a thing called peritonitis. They're very painful, and this is often a malady that we find among the elderly. So this is oftentimes why we find that we have a lot of Metamucil and fiber commercials, particularly directed at older people, so that they don't get this problem with diverticulosis and diverticulitis. Now, let's take a look at the molecule. You'll notice that the molecule is a little weird. It has a type of bond. It's an 1-to-4 glycosidic linkage. That's fairly straightforward. But since this is beta-glucose, the name of the bond is going to be beta 1-to-4 glycosidic linkage. Beta 1-to-4. You'll also notice something a little bit odd. That in the graphic you'll see that the first beta-glucose is right side up. The second one is upside down. It flipped 180 degrees. And then the next one is right side up. And the next one is upside down. And the next one is right side up. You have alternating bonds. Right? So each monomer is oriented in the opposite direction of its neighbor, which you don't see in plant starches. Right? So that's weird. Now, in this little show, they show you the flip, but I mentioned if we went back to lactose and we saw how you write the flip, oftentimes you'll see that N-shaped bond. They'll often do that with cellulose too, just to kind of show you that it flips. Now, we have one more polysaccharide I want to show you, and this is also a structural polysaccharide, and this is what we call chitin. Now, chitin is found in two different groups. The first group are the fungi. So when you look at mushrooms and yeasts, all right, and molds and smuts, all right, all those organisms, and there's about 80,000 species of fungi. They all have a cell wall, but their cell wall is not built from cellulose like a plant. It's built from chitin. Right? It's built from chitin. The other group that you're going to find it is going to be among the arthropods. The arthropods are things like insects and spiders and ticks and mites and granddaddy longlegs and scorpions. Right? Things like crustaceans. So if you look at a crab or a lobster or a shrimp, or you look at a centipede or a millipede, all those arthropods all have an exoskeleton, an external skeleton that is built from this carbohydrate. It's built of chitin. So when you accidentally step on a bug and you hear that crunch, you're breaking a carbohydrate. You're breaking sugars. So let's talk a little bit about the structure of this, and you'll realize very quickly that it looks a lot like cellulose. So, again, our monomer is going to be beta glucose. It's just beta glucose, just a long chain. You'll also notice that it doesn't branch. It doesn't branch. So just like cellulose, it's an unbranched molecule. Now, there is one difference between chitin and cellulose, and that comes at carbon number 2. Carbon number 2, if you look at carbon number 2, we would be expecting a hydroxyl group, but we don't. We have this strange little side group. This side group has a name. It's called an N-acetyl group. Now, the N tells us it's nitrogen. So, bonded to nitrogen. Right? You see that little C with a double bond to oxygen. That's part of an acetyl group. So, in this case, this is why we call it an N-acetyl group. Beautiful little side chain there. Now, you also see the term glucosamine. Oftentimes, chemists, when they have a couple of molecules joined together, will give that molecule a new name. So you have the two components, and then those two components bonded together give it a new name. Right? It's almost like, for example, we often have portmanteaus. All right. I'll give you an example. If you have when Brad Pitt and Angelina Jolie, all right, they together would be Brangelina. All right, so you see how we combine those two names? Or, for example, if we have Ben Affleck and Jennifer Gardner, they call it Benifer. And my favorite is, if you're a Big Bang Theory fan, Sheldon Cooper and Amy Farrah Fowler, Shamey. That's a portmanteau. In our case here, we have a beta-glucose molecule and this N-acetyl group. And if you notice, you've got a little nitrogen combined to a hydrogen. That's very similar to an amino group or amine. So they put those two words together to call it a glucosamine. Glucosamine is basically just the beta-glucose bonded to that N-acetyl group. So that's what you're seeing there. You'll notice that the bond between the monomers is exactly the same we saw in cellulose. This is a beta 1 to 4 glycosidic linkage. A beta 1 to 4 glycosidic linkage. Now, I mentioned that there are some polysaccharides that are very short. And it turns out these are particularly rich in certain types of plants, particularly in the legumes, which are things like beans. And we call these the oligosaccharides. Oligosaccharides, oligo just means scanty or few. And of course, you know saccharides mean sugar. Right? These are found in rich amounts in the legumes inside beans. Right? So these are the ones that actually generate the flatus, the gas. Now we make normally about 500 milliliters of flatus every day. Right? So if you go and consume something that is rich in oligosaccharides, that'll increase your flatus content. Right? So the two that are found in there are one called raffinose, and you'll notice that raffinose and the other one, stachyose, are very similar. The big difference is the number of galactose. So if you look there, you'll know with raffinose you've got an alpha glucose bonded to a fructose molecule that gives us sucrose. And then coming off the alpha glucose is a galactose. So that's kind of cool. We've got all three different types of energy-containing monosaccharides, hexosugars. Stachyose just has 2 galactose. So basically it's sucrose plus galactose, or sucrose plus galactose and galactose. Now, I will have linked to the module on carbohydrates this short little film. And it's a wonderful little film because it shows you what happens in digestion and how these 2 oligosaccharides actually lead to an increase in flatus. So how a bean becomes a fart. All right. Now that ends our lecture on carbohydrates. All right. We're going to talk about lipids next time. So I'll see you later.